The next speaker will be Professor Eörs Szathmáry, who is a biologist, a theoretical biologist, one of the best-known evolutionary biologists of our days. And I cannot speak about Eörs without some personal emotion, since he's one of my best friends. And his main contributions, together with John Maynard Smith, the two landmark books, The Major Transitions in Evolution and The Origins of Life, have been translated in more than, I think, 20 languages nowadays. Of course, he has some other famous publications. And in this regard, let me just share with you a personal story. So in 1981, 1 week after I received my MD degree in Budapest at Eötvös University, I escaped communist Hungary in order to pursue my studies in physics at Cambridge University. I had to escape because at that time there was communism. But physical chemistry was a bit boring for me, so a gloomy afternoon, I escaped even Cambridge and went to Sussex, where John Maynard Smith was at that time sitting in his little room at the end of a murky, dark corridor, and I just sneaked into his room without saying, no letters, no telephones before, and I ended up in his armchair, and we were discussing an afternoon whether I should make a PhD thesis with him or not. Unfortunately, I didn't make it for a number of reasons, but it turned out to be 10 years later or so that my best friend Eörs Szathmáry has been the main collaborator of John Maynard Smith and published these landmark pioneering books with John. So in a way, when I arrived here in Singapore and learned a little more about Chinese philosophy, I'm not surprised anymore. This is small world.
Distinguished colleagues, ladies and gentlemen, it's extremely good to be back in Singapore. And I thank Jan and Balázs for organizing this series of events. And I am going to give you some very interesting cases of evolutionary reasoning where, you know, the real power of evolutionary thinking, I hope, is going to unfold. Now, just let us remind you, you may not have seen this picture before, in the series, that in fact the thought of Darwin can be summarized on one slide. It's actually quite a remarkable thing that a theory which has so much power, the basics can be summarized on one slide. But same for quantum physics, same for general relativity. So at least in the natural sciences, theories that can explain complex phenomena don't have to be complex. Social scientists don't believe in that on the whole, which is a very strange thing, but that's the case. But we do. Now, of course, once you get to explaining things in detail, then, of course, think of mechanics. Yeah? Newton's laws are very simple. But if you want to calculate whether the bridge actually breaks or not, 40 pages of calculations, right? That's exactly the same for evolutionary theory. If you want to do the sums, as John Maynard Smith would have said, then, you know, you have to do the sums and you have to go to the nitty-gritty of the situation. Now, Darwin, this petrified gentleman over there, he actually knew what is on this slide. He didn't express it exactly in those terms, but he knew that actually 20 years before The Origin of Species was published, which was published 100 years before my birth. It's irrelevant, but very funny, right? So 120 years before my birth, he already knew this. So it's very simple. And note that this is kind of a tacit algorithm, right? You have to reproduce things. You must have inheritance, A to A, B to B, and the inheritance shouldn't be exact. And if it's true that some of the hereditary traits affect survival and/or fertility, then in a population of such agents, evolution by natural selection can go on, full stop, end of the theory at its basics, right? Now, of course, you can immediately ask questions. Yeah, but aren't there quantitative criteria? heredity shouldn't be exact. But if it's too inexact, what's going to happen? Okay, we are going to get into this a little bit. So this is actually John Maynard Smith, cutting through the, I'm looking for a polite word, through the problems, right? I am going to possibly speak about the first two, and if we are very lucky, we are also going into the third one, but we shall see. So first of all, I would like to address this difficult issue of the origin of life. Now, of course, you could say that, the origin of life is not a problem for biologists, because by definition, before the origin of life, there was no living system, and before a living system, there was no evolution, which is absolutely not true. Now we know that evolution extends into the molecular domain, and probably it preceded cellular forms of life. So therefore, we can reclaim some of the territory colonized by chemists when we want to understand the origin of life. We want to have something that can sort of reproduce and can have kind of hereditary behavior, right? So from the chemical point of view, replication always rests on what is called autocatalysis. This guy here, A, catalyzes its own formation at the expense of the difference between X and Y. And then you have got 2A. Now, this is actually very important for biology. It's much more general than DNA, because it can happen at many levels, as you will see. And it's also true that things like that, that look similar in this abstract form, were also before DNA. And after DNA, things also happened that can be described in such a way. Now, here's a concrete example, ladies and gentlemen. This is the so-called formose reaction. So this is one molecular formaldehyde, and this is glycolaldehyde. And this is a very slow reaction, if at all, but this is not the interesting part. So you have got one molecule of glycolaldehyde here, spontaneously it reacts with one formaldehyde. This is already a sugar. It has a certain transformation. Then it takes up another molecule of formaldehyde, another spontaneous rearrangement, and bang, 2 molecules of glycolaldehyde. And you don't need enzymes for this process. Moreover, the guy who discovered this was Butlerov. It's 1861, slightly before the publicity stunt of Stanley Miller, right? But of course, people somehow did not catch up. Of course, he did not know at the time that that was autocatalytic. It fits exactly the picture, but it actually happens to be autocatalytic, and this is very important. Okay, so it's 1861. So that shows that if you are looking carefully, you might be able to find more of these chemical systems that already do something that biological systems can, or what is already important for biological systems. Now, I mentioned very briefly the Miller experiments, and you really want to understand how the molecular armamentarium of living systems have come about through what they call chemical evolution. Ladies and gentlemen, I have to disappoint you. There are many answers, which means that we don't know, right? That's the problem. But here I just want, it's not chemistry, the lecture, don't panic. But I just want to show you something which is remarkable. So many people had the hunch that primitive forms of synthesis of compounds that are important for biology were very similar to the pathways that you see today, the pathways that Sydney was mentioning, leading to amino acids and all kinds of things. And there was some very limited evidence for that. But, of course, people were having very difficult problems, because if you wanted to have something like the synthesis of a nucleoside, which is here is the base and here is the sugar molecule. It was easy to synthesize sugar molecules. It was easy to synthesize nucleotides. And when you put everything together, you get a mess, completely uninteresting for biology, right? So what what can you do? And this brilliant chemist, John Sutherland, said, maybe that just doesn't apply. Don't stick to contemporary biology, because chemical evolution must have been in deep time. It may have been so deep that there is no trace of many of the things, right? Because it has been, as a palimpsest, so overwritten by biological evolution, and then the contemporary chemistry may not have strong clues to what happened at the beginning. I don't know how general this is, but just to annoy you, I am putting this on the table, because what he found, that in spite of this process that happens today, but enzymes are doing it, then he figured out, and that's the genius from a chemist, that if you start from this molecule, after several steps, but with very good efficiency, you get to the buildup of a nucleoside, right? And you can see which part comes from which part of this initial molecule. Of course, you need other reactants. So that is a celebrated result, of course. But shows that chemical evolution may have been very different from contemporary pathways. Now, suppose that somehow, we don't know how, you go to the stage where you have got something like replicators, hereditary replicators, like RNA molecules and whatever, right? Which already have formed a string and you can copy them or whatever. Do we know, by the way, from a chemical point of view, how that happened? I think Jack Szostak was here in this lecture. The simple answer is we don't, right? Unfortunately, he had to retract one of his papers, that was on the way. I mean, it happened, 2 weeks ago. So that shows that, it's a very formidably complicated problem. But, okay, let me be very generous, and I assume now that somehow the replication problem from the point of view of molecular gymnastics was solved somehow. Okay, but then we have another problem. It's like this dance that it's as this in the Arab world is so. That there are several whales around the lady, and right. But in this case, when you consider these questions, it's a kind of dance when with every whale. The lady looks uglier and uglier, right? Because the problems are coming up. Okay, so here is one, which is a crucial insight by Manfred Eigen, who is a physical chemist by training, and he got his Nobel Prize in 67 or 68 for the kinetics of fast reactions. And then he said, fine, now I can allow myself to do things that really interest me. It's good to try to win Nobel Prizes. I recommend it because, then you can do whatever you want, almost. So he considered the following question. There is a problem probably with the replication accuracy, because now the replication accuracy is something like 10 to the minus 10 per nucleotide per replication. That's a very, very accurate copying process. But you have a number of enzymes busily working on this problem. But at the origin of life, at the dawn of life, that was not given. So he gave an estimate how accurate could have been the replication of early nucleic acid-like molecules without enzymes. And he came up, from physical chemistry, that what you could reliably copy would be something like about 100 nucleotides. Now, this is just one transfer RNA molecule, right, roughly. That's 77 nucleotides, but it's the same. If you want to say, well, that's one gene. Now, one gene doesn't make a genome, right? But, if it's longer, you cannot maintain the genetic conformation. Selection is unable to maintain the information. So then you have the idea, okay, fine, so don't link them. Huh? Let you have a genome in pieces. But if it is in pieces and each molecule is replicating independently, there will be, by accident, there will be faster replicators, slower replicators, and you are going to lose, members of the genome, because the fastest replicating gene is going to push out the others. So something must be done about this. This is what we call Eigen's paradox today. And then he came up with an ingenious idea, which is the molecular hypercycle. The molecular hypercycle is for biologists, it's like molecular mutualism, right? So this is a replicator, right? And it's autocatalytic because it catalyzes on formation. And superimposed on this, there is a second cycle. And then you have a complete circle. And because it's a cycle of cycles, that's the hypercycle, right? So it's a combination of autocatalytic and heterocatalytic aid. Now, as they say, it's an exercise to the reader to prove that this is indeed ecologically stable. Did we solve the problem? No, again, another whale is going to be ripped off because Maynard Smith looked at this problem in 1979 and he said, no, wait a minute, I don't understand this, right? I mean, of course, it's ecologically stable if you don't have these nasty mutants that are perfectly possible. So this is a guy who would accept the help of this member of the cycle but doesn't give any help to the others, right? There is no link. Again, it's an exercise to the reader, and then I'm not showing calculations because I know the rule of Stephen Hawking that every line of equation cuts the remaining audience in half, right? So it's an exponential decay. I'm not going to show equations, but it's very simple to show. You just have to do an ordinary differential equation to demonstrate that if this arrow is stronger than this arrow here, so if this arrow leading to a parasite is stronger, then it doesn't matter what else is there in the system, the parasite is going to kill the system, right? I'm sure that in Singapore everybody pays taxes, right? But in some other countries maybe not, right? So the parasite problem is a recurring problem at every new level of biological organization, and it has to be solved somehow. And the hypercycle is not a solution, at least not the naked hypercycle. Okay, so before I give you what kinds of resolutions we think actually work for the parasite problem, let me mention that when we are dealing with these issues, we think that we are in the so-called RNA world. And Shostak must have at least him, he must have mentioned the RNA world. The RNA world is the world in which we sort of strongly believe in. You have to be careful how you formulate because we are walking on thin ice here. Where RNA molecules were acting as enzymes as well as hereditary material. There was no division of labor, another nice concept, right, for evolution. And just because it's so beautiful, I wanted to show you this ribozyme that was found by Michael Yarus at Colorado at Boulder, you know, that this, you know, it's a 1, 2, 3, 4, 5. It's a pentanucleotide that can act as an enzyme with this guy as kind of, you know, the substrate. And then here at this end, the smallest ribozyme that carries out a complex group transfer is this sequence here. And it is acting to amino acylate this guy here. So the amino acid that is floating here somewhere in the solution is attached to this nucleotide, right? And such reactions are important, of course, for later for protein synthesis. Moreover, it can do then the attachment of a new amino acid to the previous amino acid. So it's actually what it is done is peptide synthesis. An RNA molecule of 5 nucleotides long. Okay, so these are the results that show us that there must be some truth in the RNA world idea. Okay, now, so that these are the assumptions behind the chemistry of what we are talking about. But we want to come back to the parasite problem. And one of the resolutions of the parasite problem was basically the so-called structure D model by David Wilson. And here are two types of replicators, right? Let us say that you know, these guys, the dark guys are the nice replicators, and these are the parasites, right? And we have learned that if it's just a globally mixing pool, the parasites are going to kill the system. It goes down the drain. However, if you are doing this operation, that you randomly assign molecules into these different niches or local crevices in rock or whatever, and then the interactions are happening in the local groups, then you can make the harvest, which goes into the second generation of randomly mixing global pool. You can show if it is the case that the good guys are actually feeling their own presence. This is a bit technical, but I have to say it so that if somebody is really very deep in this, that I'm not talking nonsense. So if already one guy can feel its own presence in the compartment here, then you can prove that you have a stable equilibrium between a parasite and the nice guys, yeah? It's a so-called protected polymorphism. It's a stable equilibrium in between, right, of the extremes. So if that's the case, why not to demonstrate that such a thing is possible? And that is something that we did together last year, together with a highly competent group around Andrew Griffiths in Paris. So what we were doing is that we were putting ribozymes into artificially created compartments. Yeah? It's a kind of water in oil type of artificial microfluidic system, right? And and what you are doing is actually the following. So here is the replicator ribozyme. These are certain parts for recognition by the enzyme here, and the replicator here is the potential code for the catalyst in action, and that's a ribozyme that can cleave another ribozyme, right? And so this is a bit of cheating because it's a protein, and you do the replication cycle by the protein, but the interesting action happens at the level of the RNA molecule, because this part, the negative strand, is catalytic. And if it performs the catalytic action, you can see it because a fluorescent signal lights up. Okay, so you can sort the droplets individually. You are good, you are bad, you are good, you are bad, so and so on and so forth. So, right, so what you are doing is that out of the global pool of RNA molecule, you put them into small droplets, let the reactions go, and then you look at the individual compartments. One by one you can do it, that's the fine thing. Where there was no reaction because the compartment was full of parasites, you throw it away. You know, you are implementing this effect that I was talking about. And the nice ones you are putting back into the pool, just as the Wilson model. So what you can experimentally show that under these assumptions is indeed true. There is a stable equilibrium between parasites and useful RNA molecules. You are not going to lose the useful guys. Now, that what you could call, that's a transient form of compartmentation. And the question is whether you could do it with cells, right? Now, not prokaryotes, but protocells, you know, that are already reproducing compartments. The previous compartments were not reproducing. The interactions happened in the compartments, but everything was transient, right? The question is whether you are doing something which is more cell-like, whether that's even more interesting. And that's the old stochastic corrector model of mine from 1987. So here you have reproducing compartments, and what you have is actually that there are, we assume that there are two types of genes that are important for the replication of the protocell, right? And if one of the compartments has a if any of the compartments has a distribution that is highly distorted, which is not healthy because there are many types of one gene and only one type of the other gene, and here there is a dosage effect, then there is selection against it. Now what you can demonstrate is that this is the crucial step because every molecule can go either that way or that way. It's a stochastic process. And the stochastic process is that is generating the variation in the composition of the compartments. But the fitness of the compartments, how fast they are, that depends exactly on that composition. And there is a optimal composition with a balanced internal. So what you do, you see, is that even though, for example, the green guys are replicating faster than the red guys, yeah, the Eigen type of problem, the selection at the level of the compartment, due to the variation that is arising here in the stochasticity, can hold this malign tendency within the compartment. And this, I mean, this is very important to say, and maybe some of you realize why I'm saying that. You have no clue about such a process if you do not do the mathematical model, right? You can talk endlessly about it, but you will never know whether it works or not. You have to calculate, what are the outcomes of your assumptions, and once you have it, then you can, of course, try to make an experiment, but experiments are very difficult to make, and this system is not implemented yet. Okay, now, so I will jump over something now, and I would like to say a few words about evolvability. How are we doing with time?
We finished in 30 minutes.
Oh, there is a nice quote. Yeah, there is a nice quote from Maynard Smith about that, but I tell it in the break. Yeah. Okay. So I want to say some words about evolvability, because this, as we are dealing with it today, it's a fairly new concept, right? Although one kind of evolvability has been researched for a long time. If you remember my first slide about the Darwinian principles, there was an interesting and important process missing, from this branching process of the reproducing agents. And that missing process was something that probably many of you very much like. What is that? Sex. There was no sex on that slide, you remember, right? The A and Bs were reproducing like bacteria or simple molecules. Now, if you put sex into the game, literally, then you will see, I'm cutting a long story short, that under certain circumstances, sex is really very good for evolutionary advancement, what we call the response to selection, because those agents that can undergo sexual recombination can respond to directional selection better, so they are evolving faster. So in that sense, sexual recombination was an early form of actually searching for the evolvability question. Yeah? What makes a population more evolvable than others? But what I want to say goes beyond that. And this goes already in the direction that Gerd will be talking about. So let's talk a little bit about modularity. Modularity, if you look around, is actually quite important for all the organisms, and you can look at it in two different ways. So you could say that, for example, an earthworm is a nice modular organism because, you have these parts of the earthworm repeated nicely, one after the other. So that's, you could call a morpho-functional type of modularity. And then you can ask the following question: how do the genes affect the organism, right? Because the effects of the genes could also be scrambled or modular, right? So imagine, right, that you have one gene, and one gene would influence your eyesight, the size of your toe, your heartbeat, and so on and so forth. Now, if most genes would be doing that, that creature would be almost unevolvable because of what we call the pleiotropic effects, right? So if the genes wouldn't be acting modularly on the phenotype of the organism, you would have something which is called the cost of complexity. The cost of complexity is that if you have an influence on the phenotype by the genes that forms basically a random network of genetic influences, that's not an evolvable type of thing. Now, okay, so with this type of introduction, I just want to tell you that if you look at Darwin's finches, you already see very nice examples of that. Actually, Darwin, when he was looking at Darwin's finches, he didn't know that it was his. That was later. The second, he didn't know that they were finches. Right? That was identified later. But he did see very clearly that on the Galapagos Islands, the finches have all kinds of adaptations to different kinds of food. And that's a beautiful example of evolution. And actually, they are evolving quite fast, right? So speciation can occur in our lifetime. So that's not true that you have to wait for speciation endlessly. But it is true that if you look at the beak, which is very important for survival, and the genetic influences, then you will see that basically here there are two types of genes that are specified. And then depending on how these genes are acting on the beak, you can actually, in the geometric space, make a combinatorial formation of the different traits that define the beak, right? So there you see that there is a kind of action of the genes that is modular. I wanted to modular in the sense that they are acting on certain, they are cutting the phenotype by the joints, as it were, if you see what I mean. Now, modularity can, of course, also happen at the level of RNA molecules. Transfer RNA looks like a modular molecule, right? It looks like this, this and this, and that's the trunk, and there is another arm. I'm not evolved enough to show this, but you know what I mean. And these are basically similar modules. And as you are heating up a molecule, then of course you can see that one after the others, the modules are annihilated. I am showing this because of the following experiment. Okay, so this is, I want to show the title. It's freely downloadable in computational biology. That's of outstanding importance. This is why I want to say. Facilitated variation, how evolution learns from past environments to generalize to new environments. This is absolutely important. It's from Uri Alon's group from Israel. And let me explain what they actually did. So what they did, it was an in silico experiment, because that's much faster. And you can do in silico experiments on the phenotypes of RNA molecules in 2 dimensions, because we have a beautiful algorithm for that. And you can download it from the Vienna package, for example, and it's going to calculate the 2-dimensional shape of an RNA molecule. So exceptionally, you have a calculatable genotype-phenotype mapping, which is, of course, something which is not very common in biology. Now, and what they did was the following. So they were selecting for different shapes by simulated evolution, right? Steps of mutation, let the guys fold, and then if the fold was close to the target, then they selected that molecule, right? Now, what they did is, look at, for example, this guy, okay? This looks very much like a transfer RNA molecule. So they were doing the selection in such a way that for certain environments, only, for example, all the 3 arms were important, so that they are there. And then they switched the selection regime to something which looks like this, right? So there, only these 2 arms remained, and so on and so forth. So they were creating a selection regime where they were selecting for the modular appearance of the targets for selection. Sometimes it was the left arm that was important and the rest didn't matter. Sometimes the right arm, sometimes all 3. But they were alternating between the selective environments. And what they did, fine, now that's the important thing. So suppose that you are doing this for a long time, right? Okay, and then you are under a selection regime, right, where the molecule looks something like this, right? And then you switch back to the regime that favors this form. And you can ask the question, how fast is going to are the molecules going to adopt this conformation that they have seen previously? And the answer is that once they have undergone these repeated cycles, when they are thrown back to a previous selection regime, they find the same phenotype much faster than they were finding it when the experiment was done in the first instance. Why is that so? That is because they have evolved an amazing phenomenon. Here are the triggers, right? So that are spontaneously evolving in the population. So they have found that these are of canonical importance because if you mutate them, you have an instantaneous transformation of the phenotype, right? And that is what indirectly they were selecting for, right? And that's kind of evolutionary learning. They have a memory, right, of what kinds of environments they have seen previously. So then, of course, if you look at these systems, they have what is called facilitated variation, right? So the variation is not as blind as it was when the experiment was beginning, but under the repeated training, understood selection, the system became more evolvable. And that more evolvability means that the variation is facilitated in certain directions that are more likely to generate good phenotypes, right? Let me show you what, in very simple terms, this means. Look at this table here. Suppose, and people are using genetic algorithms for evolutionary production of different kinds of things, right? Now, suppose that you want to generate tables that you want to sell by a genetic algorithm. You can do it. You can have a representation of the structure of what it means to be a table. Then you introduce mutations, recombinations, and whatever in the underlying genetic material. And according to the code that you have in the engineering, and must be engineers around, then you are generating a certain thing that is a potential table, right? Now comes the question. Imagine that you have a representation. That's a very key concept here. You have a representation where you would have in the algorithm the rule that each of the legs would mutate independently. And then you have a representation when the 4 legs can mutate as they wish, but only in a correlated form. Can you guess which genetic representation is going to give you more money? Of course, right? You don't want chairs like this. You cannot put anything on them, right? So that shows exactly that evolution can select for representations that are more likely to make sense under the operations of mutations and recombination. That's a new insight. That's really not in Darwin. right? That's a new thing. How many minutes? 10. Lovely. Excellent. Excellent. Thank you. Okay, so we actually do have a time for evolution in learning. Yeah? And there is also learning in evolution, which I touched upon a little bit with the facilitated variation, kind of, but that's another topic. But evolution learning, what the heck does that mean? So first of all, I have to tell you that, because not everybody is a neurobiologist here, that one of the established changes in the nervous system when you are learning something is going through the Hebb synapse. Hebb was a psychologist who postulated it, but it was found, which meant, that basically neurons that fire together wire together. So if these two, if the neurons are firing at the same time, then the link between the two neurons, the synapse, is going to be strengthened. It's a very simple rule, and it's known to exist even. I just have to say it because it might be important in what I'm going to say. But before we get into the mechanistic detail, when we get there, I want to call your attention something, because this is really remarkable and very important, and this will lead to something which is a real insight. So here is the Reverend Bayes, here is Mr. Charles Darwin, and there are two very interesting quotes. First, inductive inference is the only process known to us by which essentially new knowledge comes into the world. Ronald Fisher. Now, it's the same Fisher that is one of the founding fathers of population genetics and statistics. He had another career, and the foundations of statistics, right? And that's in the book, The Design of Experiments. Then, Richard Dawkins in The Blind Watchmaker says, the theory of cumulative natural selection is the only theory we know that is in principle capable of explaining the existence of organized complexity. The question is whether these two statements are two independent statements or whether they are actually the same. That's exactly the big question. And we come to an insight which I could have generated and I didn't. There is a long list, of course, that I didn't discover, but this one I am ashamed of because I could have. It's so bloody simple. Look at it, right? So there are two people, Cosma Schalisi, and in a much more transparent form, this guy Harper. It's 2010, so we are not in deep time. We are almost in recent time here. So look, here is the equation for the Bayesian update, right? So this is the prior probability of the hypothesis, right? Then you take into account of the evidence that is there, and then you have a normalization, and then you have an updated version of the hypothesis, and this is the probability of the hypothesis being correct by taking the evidence into account. One thing. Second thing, this is the discrete type replicator equation, right? Here is, which means that the frequency of a genetic variant in the next generation is proportional to its own frequency multiplied by the fitness interaction, which can depend on many things, right? Then is divided by the normalized fitness, and you have an updated frequency of the genetic variant. It's obvious to see that formalistically they are equivalent, and here is the translation table. In the Bayesian inference, prior distribution corresponds to the population state of the evolving population. New evidence comes from the fitness landscape. Yeah? How good are you as an organism? Every organism submitted to the environment is a new hypothesis. Whether you can live well in the environment or you cannot, right? And then you have a normalization, which is mean fitness. And then you have the posterior distribution of the hypothesis. And you have the population state in the next generation. Right? So you could actually argue, and this is important, that, and you know that Bayesian learning theory is a very strong field in cognitive psychology today. And it is actually producing much more exciting results than deep learning. Because deep learning is very good for machines. But you are actually most of the time not learning deeply, right? Because you are not seeing 10 million examples in 4 hours and these kinds of things, right? Deep learning is very good for technical things, but not for describing what happens in us. The Bayesian picture is much closer to it, and there is evidence for it. But the kurz und gut, as the Germans would say, what we should remember now that if you see a Bayesian process, you can always ask the question whether the sampling process that is doing the Bayesian inference, whether that sampling process is actually identical to a replicating population. It's a valid question. There are various ways to approximate, the posterior distribution by sampling. But many of the samplings that they are using technically have the problem that they can easily get stuck, on a so-called local hill or close to a locally optimal case. That does not happen in an evolving population if you have recombination in the system, right? So that's one of the things why you might think that if you see something like Bayesian behavior in a brain, you might actually ask whether, especially in the complex situation, the underlying sampling process is a Darwinian process in real time and possibly at the millisecond time scale. That's the question. Now, the idea that selection is important for the brain, selection as such is not new. Our common friend Jean-Pierre Changeux, by the way, also the discoverer of allosteric regulation. You remember Sidney mentioned allosteric regulation. So he did actually posit, and he is right, this is a valid process, that as the organism develops early, then there's an overproduction of neurons and connections, and there is the selective pruning away, which doesn't fit. But you will see, as it is portrayed, this is a one-shot game, right? It's overproduction, you eliminate, and that's it, right? And it has a limitation, right? Because if that's all what we see, then this is the transiently redundant population. And then either you have the solution in the transient redundancy or you don't have it there. In the original publication, he explicitly says an organism cannot learn more than is initially present in its pre-representations. And that is what is completely unbelievable for complex thinking, for example, right? We could not do complex thinking with this picture for several reasons. Now, I am going to show you the last slide because I don't want to go into the technical details, but one of my actually probably the most exciting and of course the riskiest research project that I'm pursuing is whether you can go significantly beyond this picture so that it ceases to be a one-shot game, and whether in fact you could have something which is the breeding of ideas within the brain in real time, as if you were breeding horses in real time. But for horses, you are on a yearly time scale, and for your thoughts, you are on the milliseconds time scale. If it's evolution, then you can have hell of a lot of generations, even in a week, right? So the power of this process can be enormously strong. So models are already being developed and partly tested and so on. They are encouraging. I just want to show you one picture here, right? And with that, I would like to finish. That's not the theory, that's just a hint so that you somehow feel the atmosphere, right? Here, you have got two systems side by side. This is the molecular system. This is the so-called select system. Now, what you are doing is the following, that here you have a system where you can actually doing the selection, okay? So here you are replicating something like DNA or RNA molecules, nucleic acids that you can replicate by PCR or whatever. And then you see whether they are able to bind to certain target molecules in an affinity chromatography column, right? And those guys that bind well are going to be kept. The guys who are not binding well are being thrown away, and then you do the second round of selection, right? So it's a continuous generation of variation and selection, and you can do it as many times as you wish. Actually, it can be surprisingly fast. So with a molecular pool of 10 to the 13, you can have 6 or 7 generations, and you have a new ribozyme. So this is bloody powerful. So it has got two components, right? That it goes according to the Darwinian principle, right? And it's massively parallel. It's massively parallel because you have many molecules, and you have got many potential binding sites. Now, if you see that first, and then you see a picture, you know, that is about the evaluation loop in the brain, you know, from the cortex to, then you go to the striatum, the basal ganglia, the thalamus, and then back to the cortex, right? Now, people always say that the brain is a massively parallel instrument, right? But whenever you look at models of action selection, they always want to do it sort of one at a time. It doesn't make any sense. If it wants to be efficient, that's the wrong way to do, right? And actually, the structure of the loop allows you to do something similar on, for example, neuronal activity pattern that the selects is doing on patterns of molecular conformations. So then, if you apply this loop, and you can work out, okay, there are equations and whatever, I don't want to go into it because that will be another lecture, but there are encouraging results that it could be a valid mechanism beyond the other mechanisms that we don't deny, of course. Nobody denies other mechanisms. But if you think about complex thinking, like the acquisition of language, or working on general relativity theory for 10 years, or these kinds of, or insight problems, right, where you obviously don't have a rough and ready solution, because with the rough and ready solutions, you always get into an impasse. This is why you need an insight. So with these kinds of cognitively effortful, very effortful tasks, it might be the case that in fact something like a proper evolutionary search is going on in the brain in real time. And the question is whether that's the case or not, but I don't know. Maybe not at all. But this is something that I want to find out, because if the answer is yes, that's a very important thing, I believe. It's a new algorithmic component of high-level cognition. Thank you very much for your attention.